Hello students, this is part 5 of question bank. It includes 25 important questions from chapter number 11, Biotechnology, Principles and Processes. Question number 1, name the technique that is used to alter the chemistry of genetic material, DNA or RNA to obtain the desired result. It is genetic engineering. This technique is used to alter the chemistry of genetic material, and then this genetic material is introduced into the host organism, and thus it changes the phenotype of the host organism. Question number 2. Name the specific sequence of DNA in plasmid that the gene of interest ligates with to enable it to replicate. It is ORI or origin of replication. Origin of replication is must for replication of gene of interest ligated with the plasmid. Question number 3. In the year 1963, two enzymes responsible for restricting growth of bacteriophage in E. coli were isolated. How did the enzyme act to restrict the growth of bacteriophage? Two enzymes जो bacteriophage की growth को restrict करते हैं in E. coli, they are number 1 exonucleases which add methyl group to DNA, and number 2 is endonuclease which cut the DNA at specific points within the DNA. Now question number 4. Name two commonly used vector for genetic engineering. Two vectors, or you can say taxi, used in genetic engineering are number 1 plasmids and number 2 bacteriophage. Question 5 part A. Name the material used as matrix in gel electrophoresis. DNA को रिस्ट्रिक्शन एंडोन्यूक्लियस से कट करने के बाद DNA फ्रेगमेंट्स को उनके साइज के अकॉर्डिंगली जेल इलेक्ट्रोफोरेसिस टेक्निक से सेपरेट किया जाता है, एंड इन दिस टेक्निक Agarose is used as matrix which provide the sieving effect. Part B. Why do DNA fragments move towards anode during gel electrophoresis? DNA fragments negatively charged होते हैं, इसलिए they move towards positive electrode or towards anode. Part C of the question. What is the role of ethedium bromide during gel electrophoresis? Ethedium bromide is used as a stain which helps to view the separated DNA bands when they are exposed to ultraviolet light. Question 6 part A. Why is it essential to have selectable markers in the cloning vector? Selectable markers की ज़रूरत इसलिए है cloning vectors में ताकि हम identify कर सकें recombinants को from non-recombinants type. Part B of the question. Why are antibiotic resistant genes used as markers in E. coli? Antibiotic resistant genes are used as markers in E. coli because normal E. coli cells do not have resistance to any such antibiotics. Question 7 part A. Name two antibiotic resistant genes in the PBR322 of E. coli plasmid. Ampicillin and tetracycline resistant genes are present in PBR322 of E. coli plasmid. Part B of the question. State what happens when an align gene is ligated at SAL1 site of PBR322. SAL1 site tetracycline resistant genes पर present होती है, और अगर हम foreign gene को SAL1 site पर ligate कर दें, then transformants loses the tetracycline resistant gene and they are no longer resistant to tetracycline. C part of the question. State what happens when align gene is ligated at PVU1 site of PBR322 plasmid. PVU1 site is located within ampicillin resistant genes, इसलिए इस साइट पर फॉरेन जीन लाइगेट करने पर ट्रांसफॉर्मेंट्स एम्पिसिलिन रेजिस्टेंट जीन को लूज कर देते हैं. क्वेश्चन 8. हाउ इज एग्रोबैक्टीरियम tumefaciens एबल टू ट्रांसफॉर्म अ नॉर्मल प्लांट सेल इनटू ट्यूमर? जब ये बैक्टीरिया प्लांट सेल को इन्फेक्ट करता है, देन इट डिलीवर्स अ पार्ट ऑफ इट्स DNA कॉल्ड tDNA और ट्यूमर इंड्यूसिंग प्लाज्मिड और TI प्लाज्मिड, जिसके कारण प्लांट सेल ट्रांसफॉर्म हो जाता है इनटू ट्यूमर सेल्स। क्वेश्चन 9, हाउ कैन रेट्रोवायरस बी यूज्ड efficiently in biotechnology experiments in spite of them being disease causing? रेट्रोवायरस में एबिलिटी होती है कि वह एनिमल सेल को कैंसर सेल में ट्रांसफॉर्म कर देते हैं, लेकिन अगर इस वायरस को डिसआर्म्ड कर दिया जाए, इसमें प्रेजेंट डिजीज कॉजिंग जीन को इनएक्टिवेट कर दिया जाए, इवन देन it has ability to deliver a recombinant DNA into animal cell and can be used as vectors in biotechnology efficiently. Question 10 Part A, why must bacterial cells be first made competent in recombinant DNA technology and how is this process carried out? DNA एक हाइड्रोफोबिक मॉलिक्यूल है जो सेल मेम्ब्रेन में से पास नहीं हो सकता। बैक्टीरिया प्लाज्मिड को ले पाएं, इसके लिए बैक्टीरिया को पहले कॉम्पिटेंट बनाना पड़ता है एंड दे आर मेड कॉम्पिटेंट बाय ट्रीटिंग देम विथ स्पेसिफिक कंसंट्रेशन ऑफ डाइवेलेंट केटाइन सच एज कैल्शियम, जिससे बैक्टीरिया की एफिशिएंसी इंक्रीज हो जाती है और डीएनए उसमें एंटर इजीली कर पाता है। बी पार्ट, name the methods by which an aligned DNA can be made to enter number 1 a plant cell and number 2 an animal cell. DNA को प्लांट सेल में इंट्रोड्यूस करने के लिए बायोलिस्टिक गन और जीन गन का यूज किया जाता है, while in animal cell micro injection is used to introduce recombinant DNA directly into the nucleus of an animal cell. Question 11. Define transgene. It is a foreign gene that is inserted in an organism to modify its expression. Question 12. What is EcoR1? How it differs from exonuclease? EcoR1 एक restriction endonuclease enzyme है which is obtained from bacteria Escherichia coli RY13. It is used to cut DNA at a particular specific site present within the DNA, जबकि एक्सोन्यूक्लियस DNA को एंड से काटता है and remove the nucleotides or bases from the ends of DNA strand. Question 13. Explain palindromic nucleotide sequence with the help of an example. DNA पर प्रेजेंट बेस पेयर्स का सीक्वेंस जो रीड करने पर दोनों DNA के स्ट्रैंड्स में सेम हो, when orientation of reading is kept same. For example, दिया गया पैलिंड्रोम रीड कीजिए from 5 prime to 3 prime end on both the strands. ऊपर के स्ट्रैंड पर it is GAA-TTC from 5 prime to 3 prime end और लोअर स्ट्रैंड को देखिए it is same, that is GAA-TTC when it is read from 5 prime to 3 prime end. This type of sequence is called as palindromic sequence. Question 14. How is insertional inactivation of an enzyme used as selectable marker to differentiate recombinants from non-recombinants? बीटा गैलेक्टोसाइडेज एंजाइम के कोडिंग सीक्वेंस पर जब हम फॉरेन डीएनए को लाइगेट कर देते हैं, देन इट बिकम्स रिकॉम्बिनेंट एंड एंजाइम गेट्स इनएक्टिवेटेड. रिकॉम्बिनेंट क्रोमोजेनिक सबस्ट्रेट के साथ एक्स गैल के साथ ब्लू कलर नहीं देता क्योंकि इट इज़ नॉट एबल टू फॉर्म बीटा गैलेक्टोसाइडेज एंजाइम, जबकि नॉन रिकॉम्बिनेंट टाइप जो है दे कैन गिव ब्लू कलर विद एक्स गैल chromogenic substrate. So, we can differentiate recombinants from non-recombinants. Since the insert inactivates the enzyme beta galactosidase, this method is called as insertional inactivation. Question 15 Part A. Name the enzymes used to release DNA from number 1 bacterial cell, number 2 plant cell and number 3 fungus. Enzyme used to release DNA from bacteria is lysozyme. From plant cell it is cellulase and from fungus it is chitinase. Part B of the question. Identify the step in DNA extraction shown in the figure. This step is called as spooling. Question 16. Who was the first to construct recombinant DNA? Name the bacterium whose plasmid was used to cut the specific gene or DNA. Stanley Cohen and Herbert Boyer constructed first recombinant DNA in 1972. They used the plasmid of bacteria Salmonella typhimurium and isolate the antibiotic resistant gene by cutting out a piece of DNA from plasmid responsible for antibiotic resistance. And it was possible with the help of enzyme restriction endonuclease. Question 17. Study the linkage of DNA fragments shown in the figure and answer. Part 1. Name a.DNA and b.DNA. A.DNA is foreign DNA and b.DNA is a vector DNA. Part 2. Name the restriction endonuclease that recognizes the palindrome. The restriction endonuclease used is EcoR1. Part 3. What are sticky ends? Name the enzyme that link them. DNA को कट किया जाता है with restriction endonuclease enzyme. It is done little away from the center of palindrome और दोनों स्ट्रैंड्स पर सेम बेसिस के बीच में कट करने के बाद सिंगल स्ट्रैंडेड पोर्शंस आर लेफ्ट एट द एंड्स. ये दोनों सिंगल स्ट्रैंडेड डीएनए के स्ट्रेचेस एट द एंड्स आर कॉल्ड एज स्टिकी एंड्स. These two sticky ends are ligated together by enzyme DNA ligase. Question 18. What is PCR used for? Name the scientist who developed the PCR technique. PCR is polymerase chain reaction used to amplify gene, or you can say used to make copies of a gene or a DNA segment. It was developed by K. Mullis. Question 19. Mention the number of primers required in each cycle of polymerase chain reaction. Write the role of primers and DNA polymerase in PCR. See the diagram. Two primers are used, number 1 and number 2, in each cycle of PCR. Denaturation step के बाद जब DNA के दोनों strands separate हो जाते हैं, then two primers are added and they join with each strand of DNA at their complementary base sequence. These primers help in extension of new strand of DNA, जिसके लिए DNA polymerase enzyme का यूज किया जाता है. Question 20. Give the characteristic feature of the source organism of the DNA polymerase used in PCR. टैग पॉलीमरेज का यूज किया जाता है जिसका सोर्स ऑर्गेनिज्म है थर्मस एक्वेटिकस. टैग पॉलीमरेज एक थर्मोस्टेबल एंजाइम है जो डीनेचुरेशन स्टेप के दौरान यूज किया गया हाई टेम्परेचर को टॉलरेट कर सकता है एंड हेल्प इन द एक्सटेंशन ऑफ प्राइमर्स. क्वेश्चन 21. What are bioreactors? List 5 growth conditions that a bioreactor provides for obtaining the desired products. Bioreactors are large vessels in which raw materials are biologically converted into specific products in large quantities. Bioreactors provide optimal conditions of pH, substrate concentration, vitamins, temperature and oxygen for getting the desired product. Question 22. What is the advantage of bubbling air into sparged-tank bioreactor? In sparged-tank bioreactor, sterile air bubbles are sparged which dramatically increase the surface area for oxygen transfer. Question 23. Name 2 processes involved in downstream processing. Product के तैयार होने के बाद it is subjected to series of processes before marketing like separation and purification, which are collectively called as downstream processing. So, two processes involved in downstream processing are separation and purification. Question 24. Draw a label diagram of simple stirred tank bioreactor. Draw the diagram as shown in the figure. Question 25. Write the role of ROP gene in PBR322. ROP gene in the E. coli vector PBR322 codes for enzyme or proteins involved in the replication of the plasmid. So, that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.